Good day, students. You are welcome to today's lecture video on qualitative tests for carbohydrates. What we are going to be discussing here, we are not going to talk about the procedures because you have already done that in the laboratory. So here, basically what we will discuss is the principle regarding each test. Each of the tests that we are going to discuss, we are going to strictly discuss the principle and the reagents that are involved in the test, and also the reaction, and also the observation of colors that will be seen at the end of the experiment. So let's quickly make a recap of what qualitative and quantitative tests are. When we say qualitative tests, it's actually trying to identify or to detect the presence or absence of a particular analyte. And usually there is going to be observation of color. So you observe color after the reaction to see whether that analyte that you are interested in is present or absent. So here you are not after the quantity of the analyte. It's just to test or to detect whether that analyte is present or not, as simple as that. So for qualitative tests, it usually measures the quantity of the substance. So you are interested here in actually determining the concentration or the amount of substance of the analyte in the test sample. So here you are going to measure and quantify the substance, and it involves numerical results. So you have to get readings from after you must have carried out the analysis. So that's basically what distinguishes the qualitative and quantitative tests. So for carbohydrates, let's quickly look at the classification of carbohydrates. Carbohydrates can be classified based on the number of carbon the carbohydrate contains. So we can have those with 3 carbon as triose, 4 carbon, tetrose, pentose, hexose, etc. And also carbohydrate can be classified based on the number of sugar units it contains. So here we can basically have monosaccharides. Monosaccharide contains single sugar unit. Examples include glucose, fructose, and so on. Disaccharides consist of 2 sugar units, 2 monomeric units condenses together to form disaccharides. Here we can have lactose, which is a glucose and galactose that condense to form. We have maltose, which is a 2 molecule of glucose that condense to form that. So we have oligosaccharides, which contain less than 10 sugar units, and we can have polysaccharides, which are actually polymer of carbohydrate, polymer of monomeric units, polymer of glucose, and so on and so forth. So we can have starch here, we can have glycogen, and we can have cellulose. So, and also based on functional group, we can classify carbohydrates as ketoses, those that have ketone functional group, and aldoses, those that have aldehyde functional group. So for the qualitative test, there are many qualitative tests, but for the purpose of our lab, we are going to discuss basically 6. We'll talk about Molish test, which is a general test for carbohydrates. We'll talk about Antron test, which is also another general test for carbohydrates. We'll talk about iodine test for glycans, that's starch and glycogen. We'll talk about Barfoot test to distinguish between monosaccharides from reducing disaccharide, Selwanov test for ketosis, and Bell test for pentosis. So for Molish tests, basically we'll be discussing about the principle, the reaction, and the observation of the color change that we observe at the end of the day. So for Molish tests, carbohydrate solution is actually, that is the principle now we are trying to explain. So if you have a carbohydrate solution, it's treated with concentrated sulfuric acid. So the dehydrating agent here is concentrated sulfuric acid, H2SO4, that is tetraoxosulfate acid. Now, when you have a sugar solution or carbohydrate solution and you treat it with concentrated H2SO4, it's going to hydrolyze the glycosides to monosaccharides. And after the monosaccharide is being produced, and it is going to undergo dehydration to give what we call fruffural derivative. So after you get the fruffural derivative also, it is going to combine with sulfonated alpha naphthol to give purple complex. So at the end of the day, you are going to see a purple complex if uh, carbohydrates, the sample you are dealing with is carbohydrates. So generally, this is for all carbohydrates. All carbohydrates can be treated with concentrated H2SO4, and it will dehydrate the glycosides to yield monosaccharide, which now, in the presence of the acid, get also dehydrated to form furfural, and the furfural now condense with sulfonated alpha naphthol and to give the purple complex. So, and this our reagents, we have concentrated sulfuric acid, we have alpha naphthol, which is freshly prepared. It is 5% weight by volume in ethanol. Okay, so this is the reaction you have. For example, glucose is now dehydrated with concentrated sulfuric acid, and you have furfural derivative that is formed, which is now in this case hydroxymethyl furfural, and this in the presence of uh, alpha naphthol, you have a condensation product which is purple or violet as the case may be. And this is basically the color that you are going to observe. For negative tests, it is going to be colorless. And for positive carbohydrate sample, you have violet ring forming. So you have purple or violet ring that is formed. Okay, so let's proceed. Now for anthron, also anthron is another general test for carbohydrates. It's actually sugar unit here is dehydrated with concentrated sulfuric acid to form furfural, and the furfural now derivative will now react with anthron reagent to give blue-green complex, which is our end observation, the product that we'll observe at the end of the day. That is the color we are going to observe at the end of the day. So here, the reagents here include concentrated sulfuric acid, which is the dehydrating agent, and we have our anthron solution. So what is this anthron solution? Anthron is a tricyclic aromatic ketone. And how do we prepare this anthron solution? Anthron solution is prepared by dissolving 2 grams of the anthron in 1 liter of sulfuric acid. So what you will do here, after measuring 2 grams of the anthron, you will dissolve it in sulfuric acid and top up with the sulfuric acid to 1 liter. So this is basically the reaction here. You have glucose, which is the monomeric unit. When it's been dehydrated with sulfuric acid, you form the hydroxymethyl furfural, which now condense with the anthron reagent. This is the tricyclic aromatic ketone. This formula is easier this structure is easier to grab, and it will give you bluish or blue-green complex. So, and this is basically the reaction when you have your sample after you added your, after you dehydrated your carbohydrate sample and you have condensation with the anthron reagent, you have this color, which is bluish green or blue-green complex. So for iodine tests, you usually, the principle here, it's actually based on what we call iodine molecule entrapment. Here, the carbohydrates, as you know, for polysaccharide, let's say starch and glycogen, we have what we call amylose and amylopectin. For amylose, they are unbranched chain of the monosaccharide units that are found in either of the starch or glycogen, let's say, for instance, glucose. So for amylose, you have a chain, a straight line, a straight chain. A chain is of without branch. So when we talk about amylopectin, it's actually the polysaccharide, but with branching. So we have branching in amylopectin. So the amount of branching differ in from glycogen to starch. So glycogen is having more of the amylopectin than the starch. Okay, so here what is actually happening here, and you have a range of color that you will observe at the end of the day, ranging from pure deep blue to deep purple, and that is for starch. And it also depends on the proportion of the amylopectin. And I explained that this amylopectin is the branch polymer of the starch. So that is basically what you will observe. You have iodine that forms absorption complex, that is the iodine molecular entrapment with polysaccharides, and starch gives blue purple color with iodine, while glycogen reacts to form a reddish brown complex. And it is useful and convenient rapid tests to detect amylose and amylopectin and in glycogen. So the reagent here is iodine, and basically this is the color that you will observe, the range of color that you will observe. For no starch, you will not see any. There is not going to be either light or faint purple, blue, okay, or brown. No, you will not observe any of the color for non-starch material, for non-carbohydrate material. So for starch, glycogen, you can observe these varying colors: purple, blue, dark brown, okay, deep reddish brown, okay, so and so on and so forth. So let's proceed. For Barford test, is actually uh, in Barford test we are looking at a test that is similar to Pehelin's reaction, but we have not discussed Pehelin's reaction. So Pehelin's actually tried to look at the aldehyde and ketone carbohydrates that have that allosis or ketosis, and also here we have a series of reagents, like about three reagents here in Fehelin's test. We have copper sulfate, copper 2 tetraoxosulfate 6. We have potassium sodium tartrate and sodium hydroxide. So the reagent here is actually a strongly alkaline solution. So for Barfoot's test, actually the reagent, instead of being slightly alkaline, here it is the reagent is now reaction takes place in a weakly slightly acidic medium. So what actually happened here is you have copric acetate, which is copper 2 acetate, that is dissolved in acetic acid, and this solution now is used to reduce the monosaccharides that are present in the sample. And monosaccharides are more readily reduced than the saccharides. So it is based on the reduction of copper 2 acetate to copper 1 oxide, which forms brick red precipitate. So here, if we look at our reagent, we have Barford's reagent, which is prepared by dissolving 13.3 grams of copper acetate in 200 ml of water. And after that, we add 1.8 ml of glacial acetic acid to it. So this is our Barford reagent. And now the reaction is as follows. What is actually happening here? Where you have your copric acid or copric acetate will now that is when you are preparing the reagent, this is what will happen. Cupric acetate plus water to give you acetic acid. And then the acetic acid will now combine with cupric hydroxide, copper 2 hydroxide, and to form what we call, uh, 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 then after that, the copper 2 hydroxide or cupric hydroxide will now disintegrate to give you copper 2 oxide plus water. So here, what is actually happening when you have your carbohydrate solution, it is the copper 2 oxide that will now uh, react to give you the D-gluconic acid, which is a reduction product. Rather, it is now this monosaccharide now it is now reduced, okay, and you have the cuprous oxide, which is now the red precipitate that you see. So anytime you have reaction with copper 2 or copper 1 oxide that is formed, which is cuprous oxide, now you have this red precipitate, and this is what is actually happening. You will see red precipitate is formed, and we have monosaccharides that are reacting faster than the corresponding disaccharides. And for a solution that is having no carbohydrates, we will have a solution that is blue. So we'll retain our initial color without forming the red precipitate. So for Selvanov reaction, it is actually a test that is used to distinguish between allonosis and ketosis. Here, it is also similar to Molish reaction or Molish test, but ketosis are dehydrated more readily than aldoses to produce the furfural derivative. Now, it is the furfural derivative that is then condensed with resorcinol to form a red complex. And here, the dehydrating agent is concentrated hydrochloric acid. So you should note that this test is not entirely specific, as prolonged heating of disaccharides and other monosaccharides will eventually produce color. So Selvanov reagent is 0.05% weight by volume, resorcinol in hydrochloric acid, and this is our reaction when you have your fructose, which is now a ketose carbohydrates having ketone functional group, will now dehydrated when it undergoes dehydration with HCl to give hydroxymethyl furfural, and it's now the furfural that will condense with resorcinol to form the condensation product, which is the red precipitate that is observed, red complex that is observed. And this is the observation we have for a negative test. You retain your initial color, and for a test that is having a positive, we can have our red precipitate of residue to show that ketosis are present. So for Bell's test, it is used to in determination of pentose sugar. It is also similar to Molish reaction. When pentoses are heated with concentrated sulfuric acid, we have, when pentoses are heated with concentrated hydrochloric acid, the furfural is formed, which is now condensing with ursinol in the presence of iron 2 ion, which is ferric ions, to give blue-green complex. Okay? So the dehydrating agent in Bell's reaction is HCl, okay? Hydrochloric acid. And this test, you should note that it's not absolutely specific, as prolonged heating of hexosis derivatives will also give you furfural, and this furfural can react with your ursinol reagent to give you positive test. So you should take note of that. Now our reagent is arsenal. We have conc HCl and ferric chloride. Now this is our reaction. We have ribose, which is a 5-carbon sugar, a pentose dehydrating agent HCl, and you have your furfural, which now condense with arsenal to give you blue-green condensation product or complex. And this is what you will observe. You have blue-green complex at the end of the day as your observation if pentose is present. So this is the end of our lecture for today. Thank you for watching. I hope this really helps. And if that helps, please kindly subscribe and put on notification bell so that whenever we upload a new video, you will be equally notified. Thank you. See you next. Bye-bye.